Good morning. Today we will be dealing with chapter number 9, Economic Importance of Bacteria and Fungi. In this also we will be dealing with the A part in detail. Okay? Economic importance of bacteria. As you all know, bacteria is a unicellular organism. Okay? It is also prokaryote. When I say unicellular, that means it is having a single cell. When I say prokaryotic, that means the nucleus cell is not well defined. What I want to say is, the genetic material, it is scattered in the cytoplasm like this, okay? Normally when you see a cell, okay, in that you will see that it has all the genetic material, it is enclosed in the nucleus with a nuclear membrane covering. Whereas in prokaryotic bacteria, it is not having any nuclear membrane, the genetic material is completely scattered. Now, the bacteria, it belongs to kingdom Monera. In the earlier days, it was kept in the kingdom Plantae. The reason for keeping bacteria in kingdom Plantae was because it had cell wall. Okay. Later on, when they studied about bacteria in detail, bacteria was then kept in kingdom Monera on two conditions. First of all is the bacteria did not have chlorophyll. Okay, like plants, therefore it was removed from Plantae. Second is the cell wall of bacteria that is not made up of cellulose. It is made up of another substance called as peptidoglycan. And the third reason was, in bacteria, the mode of nutrition, okay? The mode of nutrition in bacteria was heterotrophic, whereas in plants it is autotrophic. About the size, okay? Now, the bacteria are the smallest organisms found on the earth. So, the size will be measured, it is measured in micrometers, okay? Next, you will read about the shape of the bacteria. The bacteria are commonly found in four shapes. First one is cocci. Cocci means spherical bacteria. Okay. Next is bacilli. Bacilli means the bacteria having rod-shaped body, cylindrical body. Third is spirula. Spirula means it has either spring-like or twisted body. Vibrio. Vibrio means comma-shaped or comma-shaped bacteria. Next we will move on to another part called as structure. So whenever we are learning about bacteria, you should know the structure in detail. Okay? Now, here I have mentioned the three important things, that is flagellum, capsule and plasmid. These are the new terms which you have never studied in a cell till now. So here I will tell you about in detail about the structure of bacteria. Let's start from here. This is a bacterial cell. It is unicellular, so it is having only one cell. I have drawn it here. Okay. First is glycogen granules. You know all the cells, they store the food in the form of some kind of granules. In bacteria, the food is stored in the form of glycogen granules, which I have shown you. Next thing is this dot-like structure here represents the cytoplasm, the fluid inside the cell. Third, you will see here circular DNA. As I already mentioned, that because bacteria it is prokaryotic, it will not have a definite nucleus nor a nuclear membrane. Therefore, the genetic material, which is a circular DNA, also called as plasmid here, okay? It is scattered. The circular DNA here is attached to on one side it is attached to the cell membrane. Next, another structure you have here is the cell membrane, the covering of the cell. Cell membrane is also covered by another structure that provides it a rigid structure, that is a cell wall. Here, the cell wall in bacteria is made up of a substance called as peptidoglycan. You have earlier studied about cell wall only in case of plants. In plants, the cell wall is made up of cellulose, whereas in bacteria, mark the difference, this cell wall is made up of peptidoglycan. Next, outer covering which you see here is the capsule. Okay, outside the cell, another protective covering the bacteria has is called as capsule. The capsule is basically secreted by the cell wall. It is slimy and protective in nature. Slimy means it will be a mucilaginous structure. Next you see here is the flagellum. Flagellum is nothing. It is a locomotory organ in case of bacteria, and the flagellum, it is an extension of the cell membrane. That's all. Next we will learn about the movement. Okay, now the basic locomotory organ we see in bacteria is the flagellum, and it can be used only in the liquid medium, means the watery medium. Okay, most of the bacteria cannot move about by their own effort, and they are only passively transported either by wind, water or contact. Next is nutrition. The mode of nutrition in case of Heterotrophic means the bacteria cannot produce its own food like plants. Okay, it has to obtain food from the other sources. Now, in heterotrophic also, you will see that there are two types. One is saprophytic, one is parasitic. Saprophytic means the bacteria is obtaining its nutrition from the dead and decaying organisms. Whereas in the parasitic mode, the bacteria is obtaining its food from its living host. It will be present inside a human body or an animal body and it will be deriving the nutrition from there. Next is respiration. Now the bacteria can respire either aerobically or anaerobically. Okay? So if oxygen is present, then it will breathe aerobically. If oxygen is not available, basically inside of the body, okay, like in your intestine and all those areas, then it will breathe anaerobically. Next we will be learning about reproduction. All of you know that the reproduction rate or multiplication rate of bacteria is really, it's very fast. Okay, it multiplies and it reproduces really fast. Now, by which method? Reproduction in bacteria occurs only by asexual method, either by fission or by cell division. Here what happens is, this is the bacterial cell, this is a cell wall, cell membrane, and this is the genetic material also called as the plasmid or circular DNA of bacteria. Okay. Now, this circular DNA, it will duplicate. Okay, that means it will make its own carbon copy here. After that, what happens is, the cell will expand. Okay, the cell has expanded, it had grown in size. Okay, then both of this DNA, one is the original DNA, one is the copied DNA, they will be moving to each ends, and then the cell will start constricting from the middle, from periphery to the middle, and then finally it will split into two bacterial cells. This is how the reproduction happens in bacteria.